And then I had someone help me figure out how to do this audio, so this audio should work too. So we'll do an overview of gene regulation. Okay, so the first thing that you guys are going to have to know is that prokaryotes are going to regulate their genes in different ways than eukaryotes. But regardless of prokaryotes or eukaryotes, we know that there's a lot of genetic information, there's a lot of DNA that isn't necessarily a gene. But a lot of that is genetic or DNA information that controls the expression of the gene, or the gene's regulation. So, in prokaryotes, most of the time that regulation is controlled by operons. And prokaryotes are always simpler than eukaryotes. But normally, these operons all control, this happens when there's a group of genes and they're all regulated together. And because of that, usually those genes all work together on a common process. So let's say we have so we start with some like precursor molecule, and we want to make molecule 4. Each of these steps is catalyzed by a different enzyme. So like enzyme A would help catalyze a reaction where molecule 1 could become molecule 2. Like if we're doing it like really simplistically, like this could be like molecule 1, and then we would have to catalyze a reaction where molecule 1 binds to another one to create molecule 2. And then we would need a different enzyme to catalyze reaction from 2 to 3. And then a third enzyme to catalyze a reaction where we get molecule 4. And we finally have like our final product. So this whole process where we have a beginning precursor molecule and then we have our final product at the end, this is a metabolic pathway. And a metabolic pathway is just a series of reactions that all line up together, and they're all catalyzed by different enzymes. So this pathway that I've created is an anabolic metabolic pathway because I'm building bigger molecules. So it could be something like photosynthesis, because it's building a bigger molecule. But other pathways might be catabolic, where we're breaking things down into smaller pieces. For a bacteria cell, if this is our process, it makes sense to make these 3 enzymes all at the same time because they all work together to form one task, which is making this final molecule. So there will be a gene for enzyme A, and there'll be another gene for enzyme B, and a gene for enzyme C. And they're all going to be near each other on the bacterial chromosome. But there's this area that's not genetic because these are all genes, because they are instructions for how to make the proteins, and our enzymes are the proteins. But what always precedes a gene? Yeah, so this could be, and usually it starts with TATA, so it starts with a TAT box, but this is a promoter region. And what purpose does the promoter serve? Why is it there? Well, there's an enzyme that needs to do transcription. Brandon? Mm-hmm. So RNA polymerase needs to bind to the promoter. So if we can control where RNA polymerase binds, we control whether or not this gene, or like all of these genes, are turned on or off. So part of the, in this like promoter region, there is this section called the operator. And the operator acts like a switch. And depending on whether or not the operator is switched on or switched off, we can have transcription occur. So now, let's say upstream of this, there's no reason why RNA polymerase can't bind, go through this promoter region, because the operator is part of this promoter region, and do transcription. But we could have a repressor protein bind to the operator, and if that occurs, RNA polymerase can't go through the operator and it can't do transcription. So far, so good? So this is a protein. So there's another gene that has instructions for how to make that repressor. So I'm going to put like 2 little lines here, because that means like far away, somewhere else. There's a regulatory gene. And this section of DNA, if I go through transcription, I'll make messenger RNA. If I go through translation, I will make the repressor. So depending on whether or not the repressor is bound to the operator, this turns the operator on or off. There's 2 types of operons, though. There are inducible operons, and then there's repressible operons. Inducible operons are like the lac operon, and those are normally associated with anabolic pathways. They are normally in the off position, which means that there's normally a repressor bound, just like I drew here. But repressible operons, like the trip operon, those are normally catabolic pathways, and those are normally or you know what, I'm so sorry, I wrote this wrong. This is catabolic, and this is anabolic. Sorry about that. Yeah, catabolic is breaking down. So the lac operon makes enzymes that break down lactose. So that's a catabolic pathway. The trip operon is an operon that builds an amino acid. So this is anabolic. So this, if the repressor, when it goes through transcription and translation, is in the conformation where it will bind to the operator, then the switch will normally be off. But some repressors might form in an inactive conformation, and if that's the case, then the operon's normally on, and they will only bind to the operator if there's something like a co-repressor that would change the conformation of the bind to the operator. How about some questions on that, on those operons? Yeah.
So gene A codes the stuff to make enzyme A?
Mm-hmm. So this would be, these would all go through transcription, all go through translation. At the end, this would be enzyme A. Eventually, we go through transcription and translation to have enzyme B. Transcription and translation to have enzyme C.
Is this the only way for prokaryotes?
This is the only way that you guys need to know for the AP Bio exam on how prokaryotes regulate gene expression. The other way that prokaryotes can regulate gene expression is sometimes you can have a signal molecule, and that signal molecule will bind to a receptor protein, go through the signal transduction pathway, like we talked about yesterday, and elicit transcription of a gene. So sometimes an environmental signal can induce the transcription of a gene in prokaryotes. Anything else for those bacteria? Okay, so let's look really quickly on how eukaryotes regulate theirs. So with prokaryotes that we just talked about, we talked about how they're in an operon, which means that we can turn multiple genes on or off at the same time, but they're always genes that are related to a common task or purpose. Usually they're part of a similar metabolic pathway. Eukaryotes are a lot more complicated.